Today's topic is external fertilization in sea urchin, which are basically the echinoderms. Echinoderms are the marine invertebrates. So, sea urchin undergo external fertilization. What does it mean? That the fusion of male and female gamete do not happen inside the female body. It is happening outside the female body. For example, in water. And why they fertilize in water? Because it's very effective for mass spawning. The female release egg in water and the male release sperm in water. So the egg of the female contain the internal membrane, which is plasma membrane. Outside plasma membrane there is vitelline membrane, and outside the vitelline membrane there is egg jelly or the jelly coat, and the male release sperm. So, in water, the pH is high, which is 7.6. So, they can move anywhere in water. They are not restricted that they can move in only one direction. So, what happened to restrict that movement towards only one egg, because they are species specific, the egg release a chemical which is resect. So, the concentration around the egg of resect is high, but the concentration far from the egg will be low. So, there will be a gradient. The sperm move towards the egg. This movement is known as chemotaxis. Now the sperm is doing chemotaxis and it is moving towards egg. So what happened when resect go and bind to the its receptor on which is present on the sperm? So it activates GTP and converts it into cyclic GMP. The outside concentration outside the sperm is high, which is calcium concentration. The calcium moves inside the sperm, and due to which it gains its motility and move towards the egg. Now what happens that the egg outside membrane, which is egg jelly, it contains specific ligands, which is sulfur containing polysaccharides. This sperm contain a receptor. Now this receptor bind to those ligands and the calcium channels will open, the sodium hydrogen exchanger will be open, and the phospholipase convert IP3 to calcium. Now what happens that the sperm head contain acrosomal vesicle, it will undergo exocytosis and the content will be released. Due to which the external membrane, which is the egg jelly or the jelly coat, this get digested, and this actin filament which is present in sperm, it get polymerized and forms a acrosomal process. Draw BG, a protein. This is the protein. So it helps in the formation of this acrosomal process. Now, this acrosomal process will extend. The extension is caused by the polymerization of the globular actins. Polymerization will take place and extension of acrosomal process. Now, what happens that the binding proteins, these are the specific proteins which are initially present inside the sperm. But when this acrosomal process is formed, then they get redistributed and they then distribute themselves outside this acrosomal process. Now, when these binding proteins encounter this vitelline membrane contain receptors for these binding proteins. So when these encounter this, then the vitelline membrane will get digested. Now, there will be fusion of the sperm and egg membrane. This is the egg membrane, and we are left with the sperm membrane. Now, polymerization of actin. This is the actin which is present in sperm. It polymerizes and form a fertilization cone. Now, this fertilization is an active process. Now, it will go and bind to the egg membrane, and there will be fertilization, which is external fertilization. When this fertilization cone will be formed, it will go and bind to the egg membrane and fertilization will take place, which is combining of the female egg with the male sperm, and there will be formation of the zygote outside the female body in water. So Now what happens that only one sperm can bind to one egg at a time. So there will be prevention of polyspermy. There will be two types of prevention of polyspermy: fast block polyspermy and slow block polyspermy. Fast block polyspermy is only for 60 seconds, and slow block polyspermy is it starts after 60 seconds. So what happens that we have egg membrane and vitelline membrane. So inside egg membrane we have cortical granules. The exocytosis of the cortical granules will take place, and due to this exocytosis what will happen that there is a protein post between this egg membrane and the vitelline membrane which connects these two membranes. But these cortical granules degrade this protein post, and it also clip off the receptors which are present on the vitelline membrane. So as receptors are not present, so sperm cannot bind to this egg membrane and fertilization will not take place.